Hey everyone, welcome back to day 2 of vaccines. So today we're going to pick up right where we left off. So we've done an overview of vaccines, we went through a little bit of a history, we talked about what are some of the things that effective vaccines should be or should have, and today what we're going to do is we're actually going to get into, well, how the heck do we make vaccines? And we'll talk about all the different mechanisms, and for each mechanism I will give you a couple of examples of vaccines that are made using that mechanism. And then we will end the topic talking about SARS-CoV-2 and a little bit about some of the therapeutics and some of the vaccines that we are pursuing. So let's go ahead and start with our overarching question of how the heck do we make a vaccine? So there's lots of different mechanisms. And what we're actually going to do is we're going to draw a flow chart that is going to encompass the majority of the ways that vaccines are made. And then we will actually go through each of the pieces of the flow chart one at a time. So let's go ahead and draw a virus on here. So here is the parent virus that is virulent, and what we're going to do is we're going to talk about one way in which we can take the virus and we can actually attenuate it. So let's make it a different color to illustrate that it has been attenuated. And a virus that is attenuated really just means that it has been weakened. So it's a weakened form of the virus. And we'll talk about ways to do that. We can also take a virus and instead of attenuating it and having a weaker version of it, we can actually inactivate it. So I'm just going to draw an X over it to represent inactivation. And inactivation can be using chemicals, and we'll talk about some of the examples of chemicals that are used, and in a way, it is killing the virus. So we'll talk about what that means. We can also do a method called fractionation. And in fractionation, we essentially get all of the pieces of the virus that are broken up. And so all of these methods have been used, are still used, and we'll talk about how. But a lot of effort has gone into cloning, and oftentimes this cloning is done in bacteria. So let's draw a different color here in the chromosome to represent engineering, genetic engineering, so a piece of genome from a virus. So for cloning, oftentimes we refer to this as recombinant. We also refer to it as genetic engineering. So a lot of our vaccines are recombinant vaccines or made via genetic engineering, and there's a lot of different ways that we can do this. So for cloning, we can do expression. So oftentimes for expression, what we'll do is we'll take a specific gene from a virus, oftentimes a protein, and we will have bacteria, yeast, other cells express that protein, and then we can purify it. So we can get a subunit. So maybe we make the glycoprotein, and instead of working with the entire virus, we might never have grown the virus in the lab, but because we know the sequence, we can go ahead and do cloning, and maybe we can make a glycoprotein that can be used as a vaccine. We'll also talk about VLPs or virus-like particles, and so these are proteins, usually the capsid, that are expressed and self-assemble, so we'll talk about that. We also have what I like to refer to as nucleic acid vaccines. So our textbook calls them DNA vaccines. Many places still call them DNA vaccines. I like to call them nucleic acid vaccines because, as we'll talk about at the end of this topic, you can do a vaccine that is not DNA, that is RNA. So I like to be all-encompassing and not discriminate against my nucleic acids. And then we can also actually do something called viral vectors. And what a viral vector is instead of using the virus that we are trying to vaccinate against as a vector, we use a different virus. And so perhaps we're going to clone part of the genes into the viral vector and use that to administer the vaccine. So we're going to talk about each of these different ways to make vaccines. And before we do that, I want to show you kind of an overview of the vaccines that we have. So this is a table of viral vaccines that are licensed in the US. And I really like this table because what it does is it not only tells you the disease or the virus, but it also tells you the type of vaccine, the indications for use, and then the schedule as well. So not all of these vaccines are given to everyone. It fully depends on what you do. So for example, adenovirus is given to military recruits. Hep A is given to travelers or high-risk groups. So again, this is a really nice summary table, and many of these viruses, we won't talk about all of them, but we will talk about many of these viruses and the type of vaccine that we have against them. So what we're going to do is we're going to walk through our flow chart, and then we're going to talk more details about how each of those pieces of that flow chart that we just drew are done, and then I'll give you an example. So let's begin with attenuation. So when we have an attenuated virus, one of the things that happens is that viral replication still occurs. And what this does is this is going to stimulate the immune response. And we tend to get what we refer to as mild or inapparent disease. The reason for that is because we still have viral replication that occurs. And there's a couple of different ways that we can do viral attenuation. One of them is passage in non-human cells, and the second way is genetic engineering. So on this page, let's talk about passage through non-human cells. So we're going to use a lot of colors here. So let's start with, let's say this is our human cell. So for this human cell, we isolate a pathogenic virus. And then what we're going to do is we are going to take that pathogenic virus, and using a non-human cell, we are going to passage it through that non-human cell. And at the end, hopefully, what we get is a virus that now we have selected for a virus population that infects those non-human cells better than it infects human cells. And so the goal here, again, is that this virus no longer grows well in human cells, and it's going to elicit an immune response. And there's a lot of non-human cells that are used. So a couple of examples, in case you're wondering, are monkey cells. Sometimes we use ferret cells. So in the case of the Flumist vaccine, we actually use ferret cells to passage that through. So again, the goal here is to select for viruses that no longer grow well in human cells, but they can still elicit an immune response. So another way for us to get an attenuated vaccine, or to make an attenuated vaccine, is through genetic engineering. So for genetic engineering, let's say that we have this virion, and one of these pieces of genomes here that I've drawn has a virulence gene. So back when we talked about virulence and how we identify genes, we go through that mechanism and we identify the genes that encode for virulence. We figure out what those are, and we can do a couple of things. One thing that we can do is we can mutate the gene. So maybe we mutate it to inactivate it, or we switch it out for another gene that is not pathogenic, and so we end up with a piece of genome that is different. Alternatively, instead of mutating that gene, maybe we can just delete the gene. And so now what we're left with is a virus that is missing that gene. So other things that you can do that kind of fall in between, they're kind of an other category, if you will, is particularly for viruses with segmented genomes. So for example, flu and rotavirus, one of the things that we do is we actually mix pathogenic and non-pathogenic strains, and we let reassortment occur naturally in a cell line. And then at first we try to isolate viruses that will elicit an immune response but are non-pathogenic. So lots of different ways that we can do attenuation here. And of course, when we have attenuated viruses, there are some inherent risks. And before we talk about those, let's talk about some examples of attenuated vaccines. So one vaccine that is attenuated is the poliovirus vaccine, and this vaccine is administered orally. The measles vaccine, which is injected, is another one. And another really great example is the varicella zoster, which is chickenpox. So many of you have received this vaccine, and this is a attenuated. Other ones that we just mentioned are rotavirus, which is actually oral, and adenovirus is one that is given to military recruits that is oral, and it's an example of an attenuated. And there's actually a newer vaccine that is called Zostavax, and it's against varicella zoster or shingles. So with these vaccines, these are still viruses that are alive, if you will. They're completely capable of reproduction. So you can imagine that there are definitely some risks when it comes to these vaccines. Because these viruses are able to reproduce, we actually see shedding of the vaccine strain. And when this happens, of course, this can lead to infection of unvaccinated individuals. So this can particularly be a problem if we have a population where many individuals are unvaccinated. And of course, we can get issues with reversion to virulence. Now, this does not mean that all of a sudden the gene that you deleted is going to pop back spontaneously, but as you and I know from talking about mutations and resistance to antivirals, of course, viruses mutate every time they replicate. It's spontaneous, but you can get a mutation that spontaneously somehow allows that virus to now have a new trait that allows it to be more virulent, and now that of course is going to take over the population. So especially given the high rate of mutations associated particularly with RNA viral reproduction, we can see reversion to virulence. And this is actually that we see with polio. So we see something known as circulating vaccine derived poliovirus, which is shorthand CVDPV, and this is especially a problem in underimmunized populations. So again, there are definitely risks with attenuation, and we will talk about some of the pros and compare attenuation to some of the other methods of making vaccines at the end of this topic. So let's talk now about inactivation. So for inactivation, we consider this a killed virus vaccine. And so what that means is that we use chemicals or sometimes physical procedures, And what these do is these are going to make the virus non-infective. And so the virus itself remains intact, but it cannot infect. And so some examples are things like formalin that can be used. We can use things like detergent. We can use beta-propiolactone. So these are common chemicals that are used. And the thing with these vaccines, so for inactivated vaccines, they are actually safer for immunocompromised patients. But the problem with these is that they may require several doses. So while for an attenuated vaccine, you mount a stronger response, you typically do not need several doses. When it comes to inactivated vaccines, you will actually require several doses to get to the same amount of memory and immunity. And there's a couple of examples, and one of them is polio. So we actually have 2 vaccines for polio. So this was the first polio vaccine, and the first polio vaccine was an inactivated form of the virus. And this, of course, brought the deaths due to polio down significantly. But we had what is referred to as the Cutter incident. And so what ended up happening was over 200 kids actually developed polio due to the vaccine. And the reason for that, when they looked back to see what had happened, it was completely an accident. But the virus was not actually completely inactivated. So, of course, that is definitely a con to these inactivated vaccines. Other viruses that are inactivated for use in vaccines is the Hep A vaccine, rabies, and influenza. So for influenza or for flu, we actually, if you recall from earlier in the semester, we actually grow the influenza in an egg, so in a chicken egg, and then we will use formalin or detergent to inactivate it. And every flu shot is about a chicken egg or so. So kind of moving forward, we can also think about this idea of a viral vector. And for a viral vector, what we do is we take genes from a pathogenic virus, and those genes are then inserted into a non-pathogenic virus. And oftentimes, we actually use pox viruses a lot as vectors, and so one of the ones that we use is a vaccinia virus, and so this is our vector. So this is what those genes from the pathogenic virus are going into. So let's kind of draw a picture here to illustrate that. So let's say we've got a virion here. So this will be our pathogenic virus, and we'll draw couple of different colors here of a segmented genome. And then here is our vector virus, which is a pox virus and has that really weird wonky shape. And again, what we're going to do is we're going to clone those individual genes into the vaccinia genome. And I'm running out of room, so let's draw it over here. And so what we might now get is a pox virus that has a gene from that pathogenic virus, and now maybe it's able to express a different glycoprotein, and this is going to be now our vaccine. So again, we might not even have to grow this virus that is pathogenic in the lab. We might be able to do this all with genetic engineering and all with genomics. And so an example of this is the oral rabies vaccine. So in part 1 of this topic, I told you all about how we actually vaccinate wild animals by dropping bait into the wild, and so they eat the food that they think is a tasty snack, and then they actually bite it. And then when they do that, they self-vaccinate with the vaccine, that oral rabies vaccine that is in that bait. So, of course, as you can imagine, there have been instances where people have accidentally gotten infected with this. And so there was a woman a couple years back that her dog had actually grabbed one of these baits because they lived in an area where they were using this oral rabies vaccine due to high numbers of rabies. And the dog actually brought it back to her, and she tried to grab it from the dog's mouth, and she actually had a cut on her hand from gardening, I believe it was. And so when she grabbed the oral vaccine from the dog, some of it got into her, and so she, of course, got sick from this. But anyways, the point here is to just illustrate that with all of these different mechanisms of making vaccines, there are, of course, pros and cons to both. So this is using a viral vector. Now let's talk about subunit vaccines. And there's actually kind of two ways to make subunit vaccines. And really more than two ways. You'll see over the next couple of pages that these are all kind of subunit vaccines, but I like to kind of categorize them even more than how our textbook does, because I think it makes more sense this way. But one of the ways that you can get a subunit vaccine is you can fractionate the virus. So you can kind of think, I always like to think of Hershey and Chase, and so oftentimes using some sort of physical method to chop up that virus, if you will. The second way that we can do this is to do genetic engineering, and this genetic engineering is to express the viral proteins. And for both of these methods, what you do here is you actually immunize with the viral components and not with an intact virus. So usually the proteins that we use, or what the antigen is usually a capsid or membrane protein. So again, in the case of fractionating, think Hershey and Chase. In the case of genetic engineering to express viral proteins, typically what we will do is we will clone the gene of interest, so perhaps a gene that encodes for the glycoprotein. We will then express in culture, and again, you can use different cultures, so bacteria, yeast, other eukaryotic cells can also be used, and then we purify the protein, and we make sure that it is sterile, of course. We make sure that there aren't other things in the solution, and then we test that and see if it is a possible vaccine candidate. And so there are actually a couple of examples of subunit vaccines. So one of them is called Flublok, and you can probably guess what this is against. This is against the flu. And Flublok is actually the hemagglutinin protein. And so what we have done with this is we have genetically engineered this. And this is actually the only egg-free flu vaccine. So for people with allergies to eggs, of course, they can't get the typical flu shot, so they can get Flublok. And another really great example is actually the new shingles vaccine. So there is Shingrix, and again, this is against shingles, so against herpes zoster. And so these are some of the only successful to date genetically engineered subunit vaccines that we have, at least for humans. So it turns out, based on the data that we have, at least for Shingrix, it tends to work better than the other attenuated vaccine that we have called Zostavax, which is not usually the case. Typically, we see that attenuated vaccines actually work better to elicit an immune response than subunit vaccines, and we'll kind of get into the why here in a little bit. But it turns out that, at least for the data that we currently have, Shingrix is definitely at least better. So if, when I turn 50 in a couple of decades here, if my choices are between Zostavax and Shingrix, I personally would choose Shingrix. But again, that is just me. So anywho, I digress. Moving on away from these two methods of subunit vaccines, let's talk about virus-like particles, also known as VLPs. And VLPs are a type of subunit vaccine, but instead of just being a bunch of glycoproteins, for example, what VLPs do, and this is a type of cloning or a type of genetic engineering, is we actually take advantage of the fact that many capsids self-assemble. And so when this happens, what we have is we have capsids, but they are empty. So they look like the capsid of a virion, but there's nothing on the inside. And so what I've done here is I've actually linked a video, and believe it or not, we've actually seen this video and we've seen this image when we first started talking about the structure of viruses, what seems like forever ago in this class. And there are two really great examples, and that is hepatitis B virus and human papillomavirus vaccines. So for both of these viruses, or against both of these viruses, the vaccines that we have are these VLPs. And so for HPV, many of you have probably heard of Gardasil. And of course, we've had many iterations of Gardasil now at this point that are against different strains of HPV, but the way that they are made is very similar. And so what scientists have done is they have taken the gene that encodes for the capsid protein, they are able to clone that, and in yeast or in other cell lines, they're able to grow it, and then they purify it, and then they assemble into these really pretty VLPs. So the link that is on this page is actually a link to a scientist that did his thesis on assembly, and it's specifically on the assembly of a bacteriophage, but it does a nice job of showing you how all of the capsid proteins actually come together. So Lander does a really nice job, and I recommend watching the video. All right, so moving on, we are going to talk about nucleic acid vaccines. And oftentimes people refer to these or group them into the subunit types because the nucleic acid, of course, is a part of the typical virion. And so they say that this is a variant of the subunit protein approach. And for the nucleic acid vaccines, we typically have DNA or RNA. And of course, the DNA or RNA that we use is going to be DNA or RNA that encodes for a viral gene. And so what we will do here is we'll oftentimes have a vesicle, and the vesicle is what delivers it, and then on the inside we'll have the nucleic acid. And so of course the thought is, for example, if you are delivering RNA, that RNA gets into the cell and of course will be translated into protein, which will get folded, and then you'll have the viral protein, and then you will be able to, hopefully, your immune system will recognize that as foreign, and you'll mount your response, and it'll teach your immune system that way. If it's DNA, of course, DNA would then be transcribed to RNA and then translated to protein, and then boom, viral protein. So this is why oftentimes this is considered a variant of the subunit protein approach. And as you can imagine, this is a very challenging approach. And in terms of vaccines in humans, there are not any to my knowledge, and I tried to double check yesterday to see if I was wrong, but I could not find anything. There do not appear to be any DNA or RNA vaccines that have been approved by the FDA for use in humans. An example that I can give you is West Nile virus for horses, at least. We have seen it work in those animals. Right now, there's leads in, so this is for humans, for HIV and hepatitis C virus. And as we will see, for the current coronavirus as well. So we'll come back to that. But again, we don't have vaccines that are FDA approved yet in humans that are nucleic acid vaccines due to how challenging this approach is. So we've covered all of the different ways that we can make vaccines. I have given you several examples of ones that fall underneath each category. And while we've started to kind of talk about some of the pros and cons, we haven't fully gotten to them. So what we're going to do is we're going to group subunit and inactivated together, and then we're going to talk about those versus attenuated. So in attenuated, we talked about this idea that viral replication occurs. So that is both a pro and a con. So it's a pro in the sense that because viral replication occurs, it actually stimulates the immune response better. And so we actually see usually longer term protection from these. But a con of the viral replication occurring is that you can infect others because of the viral shedding. And another con, if you will, is that you tend to get mild symptoms. So because you have viral replication, you get those mild symptoms. Now, again, because you get the mild symptoms, it also means that your immune response is better. So, you win some, you lose some. On the other half, we've got the subunit and inactivated vaccines. So for these, they don't cause inflammation. And this is definitely a con in the sense that inflammation is a key part of the immune response. Without inflammation, we don't get more signaling that the immune system needs to do its job. So that's definitely a con. We also get, in general, poor activation of the adaptive immune response. And again, that kind of goes back to we are not getting inflammation, so we have less signals and less signaling that is happening within the immune system. And so we just don't get as good of an adaptive immune response either. Other cons tend to be a little bit more expensive. Usually all of these are injected. And kind of going back to the immune problem, we have poor antigenicity. But some pros are that it is fast, right? So it might be more expensive to develop, but it's faster. And of course, another pro is that there is no viral genome or no infectious particle. So particularly for people who are immunocompromised, this is definitely a safer alternative. And we, of course, might have viral nucleic acid, but we do not have the entire viral genome, which is why we don't get the infectious particle. So some pros and cons and kind of a summary of all the different ways to make vaccines. So with all of this, something that is a take-home message that I want to remind you all of is this idea that even if we were to eradicate every single virus from the earth, as long as the nucleotide sequence is known any virus can be recovered. So if you know the nucleotide sequence of a virus, you can synthesize that nucleic acid, you can pop it into a cell, and then boom, out will come virions. And so that is just something to keep in mind with this. And this kind of brings back the idea of first smallpox. And so there are conversations, and have been of course for years, ever since we've eradicated smallpox, with, well, should labs be required to destroy all the stocks of smallpox that exist? Because nowadays, of course, most people are not vaccinated against smallpox, and so what a great way to use a bioterrorism weapon. And so again, this kind of goes back to our take-home message here. So even if we eradicated a virus, if we know the nucleic acid sequence, any virus can be recovered at any point. Let's talk about what kind of vaccines are being made and tested for SARS-CoV-2. So if you recall from day one, we said that there are two types of vaccines. There are active and there are passive. So passive tends to be antibodies or immune cells from a donor into a patient. And so even though this article that I've screenshotted and pasted here is a week and a couple days old, this was just approved recently for a clinical trial. And so one of the things that is going on that you probably heard in the news is that doctors, scientists, they are looking for patients who have had this coronavirus and have gotten better to donate their blood so that we can take their serum. And so in this paper, again, it's a journal article, but it was a nicely written journal article, and it's actually from, if you take a look at Johns Hopkins, they have a lot of really good resources in addition to the map that we keep checking out. But one of the things that they're trying to do in this clinical trial is take these COVID-19 patients who have, of course, recovered. So we get their serum, which has the neutralizing antibodies, and there's two things we're looking at doing. The first thing is therapy. So can we give this serum to patients who are currently sick? And we have some limited data that suggests for at least COVID-19 that this seems to help and ease the symptoms and allow patients to get better quicker. And again, this is nothing new. We have been doing serum transfers for decades, and scientists have known about this for a long time. But if you recall, because this is passive, this is not long term. But they're also looking to use this serum as a method of prophylaxis. All right, so last but not least, of course, passive vaccines are great. This is a wonderful solution, for the time being until we have something better. But ideally, the golden ticket, the golden standard of course is going to be our active vaccines. So vaccines are, as we mentioned, the best defense that we have against viruses. So I checked this morning before recording this lecture to make sure that this was still up to date. So the date here says April 15th. Today is April 16th. But as of this morning, the numbers were still the same. And this is a really cool website, and I've linked it on this page. And what the Milken Institute is doing is they do a nice job of summarizing and keeping track of everything that is in the pipeline for trying to, whether it's treatments, whether it's vaccines. So they keep track of all of these things that are being done across the entire world, so all different labs and institutions. So as of today, there are currently 86 vaccines that are in the pipeline at different various stages for SARS-CoV-2. So what we're going to do is I'm actually going to open up a PDF, and the PDF actually comes directly from this page. I've also linked it to Moodle, and we'll just take a look at it really quick together and kind of look at some of the types of viruses or some of the types of vaccines that are in the pipeline and where they are at in terms of, are they in clinical trials yet, and what type of vaccine they are. All right, so here we go. Here is the website. So it looks like it actually still has not been updated yet today. So let's go ahead and click here on this View COVID-19 Tracker. So again, I actually downloaded this PDF and I posted it to Moodle for you all so you could peruse it in your own time as well. The top half of this is all different types of treatments, and these treatments actually go along with the previous PDF that I posted for topic 18. So what I'm going to do is skip through all these treatments for now and get to the vaccines. So hopefully I'm not making you all too dizzy by scrolling down. There's quite a bit of treatments. All right, so here we go. Here are the vaccines. So as you can tell, there are 86 of them. We're not going to go through all of these, but I did want to show you at least a couple. So the first one up here is DNA plasmids. So you'll see the first few actually are DNA plasmids, and right now, most of them are in the preclinical. One of them is in the clinical stage of development, and they started phase 1 in April 2020. And later this summer, we should expect to see the data. If we go down, we continue going down, we see inactivated. So one of them actually tells us that they inactivated it with formaldehyde, and most of these are in preclinical. So as you can see, majority of these are in preclinical. We also have a non-replicating viral vector and other things that we have. You can see lots of viral vectors actually. So some of them using adenovirus. Going down, we actually have a protein subunit, so a capsid-like particle. So the idea of a virus-like particle, but of course we know coronavirus doesn't assemble into an icosahedral capsid, and so it's going to assemble into its nucleocapsid that it has. You'll notice most of these, again, are in preclinical. There's only a couple that have made it to clinical. Number 21 here's an adenovirus type vector. So we've got attenuated, we've got different vectors. There's actually an influenza and vaccinia vector when I was perusing through this. There are DNA, as we saw, and there's actually an mRNA vaccine on here as well. And that was the first one that went to clinical trials, and that was Moderna Therapeutics. So let's go ahead and try to not click too many things on here, but let's go ahead and actually search Moderna Therapeutics. So here it is. So for the Moderna Therapeutics, here we've got the RNA, and it is in the clinical stage, and phase 1 started in March 2020. So this was the first one that went into clinical trials. So again, you'll notice that many of these are from all different universities, different countries. So there's definitely a huge global effort that is going towards development of a vaccine. So while we're here, we can actually go back to our tracker here. And as of, let's see, what time is it? April 16th. At 12:49 p.m., we have a little over 2 million cases, with the U.S. sitting at about 640,000 cases. We will see each other in the next lecture when we get into evolution of viruses and history of disease and viral infections. And that'll round off the semester. So I hope you all enjoyed this topic. It was a lot of fun. I actually worked on completely revamping and remaking this lecture this year. So it was a lot of fun to go through and look up what vaccines are made which way. So I hope you enjoyed it as much as I did, and I'll catch you in the next one. Bye, everyone.